Today, let us take up the third part of photosynthesis. Today I am dealing with Calvin cycle, Hatch-Slack pathway, and photorespiration. In the last class, we have studied about the light reaction or light dependent reaction. So what is the end result of light reaction? Generation of oxygen, which is released into the atmosphere, then synthesis of high energy compound ATP and also NADPH. Now, first I will take up Calvin cycle. The Calvin cycle is also known as a dark reaction. Actually, it doesn't occur in dark, but it doesn't require any light. So, it is light independent reaction. Though it requires no light, it depends on the products generated in the light reaction, that is ATP and NADPH. The dark reactions occur in the matrix of chloroplast. The matrix has all the required substances for Calvin cycle. Calvin, Benson and Basham, three scientists, they have worked out how plants synthesize glucose from carbon dioxide. For this work, Calvin received Nobel Prize in Chemistry in the year 1961, and the cycle is known after him as Calvin cycle. Calvin used unicellular green alga known as chlorella in his experiments. And he raised this chlorella on a food medium containing 14C. 14C means carbon having radioactivity. So this is radioactive carbon. The food medium on which he raised chlorella contained this 14C. Then the synthesis of various products or mostly glucose is very rapid. So what he did, after every few seconds, he killed the chlorella and tried to find out what compounds were synthesized. When he tested chlorella, just within 5 seconds, after raising on the food medium having this radioactive carbon, he found the chlorella has synthesized a stable compound or a 3-carbon stable compound, and it is phosphoglyceric acid or PGA. So within 5 seconds or in less than 5 seconds, chlorella synthesized the first stable carbon compound in the form of PGA or phosphoglyceric acid. And this is the starting point in photosynthesis. This is the starting point. And after this, many steps are involved in the production of glucose. All these steps are not prescribed for your syllabus. So let us take up a simplified version of Calvin cycle or C3 pathway. And the first stable compound produced here is a C3 compound. It is known as C3 pathway. For a long time, everyone believed that there is only one pathway, that is C3 pathway. But later on, another pathway called C4 pathway was also discovered. I will come to that later on. Then coming to the C3 pathway or Calvin cycle, for the first time, we come across use of carbon dioxide. I have told you, in the light reactions, there is no role played by the carbon dioxide. But here, carbon dioxide plays a very important role. The first step is the carbon dioxide has to be accepted. So who will accept the carbon dioxide? The carbon dioxide is accepted by a 5-carbon ketone sugar known as ribulose bisphosphate or ribulose 1,5-bisphosphate. So, in the first step, carbon dioxide combines with ribulose bisphosphate, which is also known as RUBP. And this reaction takes place in the presence of the enzyme known as Rubisco. Rubisco means ribulose bisphosphate carboxylase and oxidase enzyme. This Rubisco enzyme has two important functions. It participates in carboxylation. Carboxylation means combining carbon dioxide with RUBP, and it also participates in oxidation. So it performs two functions, carboxylation and oxidation. That's why it is known as Rubisco. C means carboxylation, O means oxidation. This enzyme is the most abundant enzyme present in the world or in the living beings. It is the most abundant enzyme. Then the reaction is like this: RUBP, ribulose bisphosphate plus carbon dioxide plus water. In the presence of the enzyme Rubisco, carboxylation takes place and two molecules of 3-phosphoglyceric acid are synthesized. We simply call them as PGA. So at the end of carboxylation, what do we get? Two molecules of 3-carbon compound, that is PGA. This is the first step in photosynthesis. Then the Calvin cycle is divided into 3 stages for our convenience. And remember, Calvin cycle occurs in all the green plants. In all the green plants. So, what are the 3 stages of Calvin cycle? This is a very simplified form. First one is carboxylation. Second one, reduction. Third one, regeneration. I already have explained about carboxylation. Carboxylation involves synthesis of the first stable carbon compound PGA from RUBP and carbon dioxide with the help of the enzyme Rubisco. So this is the first step. So in this way, in the first step, or in the carboxylation, carbon dioxide is used in the synthesis of PGA. Then second step is reduction. It involves a series of steps ending ultimately in the synthesis of glucose. For the synthesis of one glucose molecule, 6 carbon dioxide molecules are used, and it takes 6 turns of the cycle. So, in the synthesis of glucose, 6 carbon dioxide molecules are used in 6 turns. And remember, in each turn of the carbon dioxide, 2 ATP and 2 NADPH molecules are consumed. So 6 carbon dioxide molecules are used, 6 turns, and each turn 2 ATP and 2 NADPH molecules are consumed. That means, for the synthesis of one glucose molecule, 12 ATP and 12 NADPH molecules are consumed here in this reduction process. Then, the third part is regeneration. Ultimately, RUBP has to be resynthesized. If all the RUBP is exhausted, the cycle comes to an end. But it should not happen. The cycle must go on continuously. So, RUBP or ribulose bisphosphate has to be regenerated at the end of the cycle. This regeneration of RUBP, or the process of regeneration of RUBP, consumes one ATP molecule for each carbon dioxide molecule. That means 6 more ATP molecules are consumed in total for regeneration of RUBP. That means in reduction process, 12 ATP molecules are consumed, and in the regeneration process, 6 more ATP molecules are consumed. So overall, 18 ATP molecules are consumed in this process of Calvin cycle, and 12 NADPH molecules are also consumed. So ultimately, this is the final reaction of Calvin pathway. So it involves utilization of 6 carbon dioxide molecules, 18 molecules of ATP, 12 molecules of NADPH. And the product is this glucose molecule, one glucose molecule, 6 molecules of water, 18 ADP, 18 inorganic phosphate molecules, and 12 NADP. So this is the final or the end reaction of Calvin cycle. I have told you Calvin cycle occurs in all the green plants. Then coming to the next one, C4 pathway. For a long time, it was thought that there is no other pathway except C3 pathway in plants. But two scientists, namely MD Hatch and CR Slack, they have discovered a new pathway which is known as C4 pathway, or it is known after them as Hatch and Slack pathway. Here, the first stable compound, carbon compound produced is a 4C compound. So it is known as C4 pathway. C4 pathway is different from C3 pathway in many respects. C4 pathway was discovered in tropical plants. The tropical climate is very harsh. It is very hot, humid, and dry. So the plants living in tropical climate have this mechanism, C4 pathway. This is mainly found in plants belonging to the family Poaceae. These plants are sugarcane, corn, sorghum, and other plants. These are all tropical plants. The tropical plants, the leaf anatomy of these C4 plants have an anatomy, a peculiar anatomy known as cranch anatomy. The leaves of these tropical plants have an anatomy known as cranch anatomy. Cranch is a German word. Cranch means wreath, arrangement of flowers in a circular way. This is called cranch anatomy. I have drawn one figure here. In the cranch anatomy, what happens is, around the vascular bundles, this is the vascular bundles in the leaf, specialized cells are present, or the vascular bundles are surrounded by bundle sheath cells, specialized cells known as bundle sheath cells. They are arranged in one circle or around many circles. These bundle sheath cells are surrounded by mesophyll cells. The bundle sheath cells are very important. They have a thick cell wall. Then spaces are absent between these cells. They got large number of chloroplasts which are bigger in size. These bundle sheath cells are surrounded by mesophyll cells. Here I have drawn one mesophyll shell. This is the bundle sheath shell. I will explain these things just after a few seconds. So this is the special type of anatomy present in Hatch and Slack pathway or in C4 pathway. Then, how photosynthesis occurs in C4 pathway. Here, carbon dioxide is involved and the carbon dioxide combines with, not with this one, RUBP, but with phosphoenol pyruvate. Phosphoenol pyruvate, which is a 3-carbon compound. This is the 5-carbon compound, but this is only a 3-carbon compound. Before I proceed with the reaction, I want to give some more points. This is the mesophyll cell and this is the bundle sheath cell. These cells are connected by channels known as plasmodesmata, through which substances can travel in and out. Plasmodesmata. The cell wall of the mesophyll cells is thin. The cell wall of the bundle sheath cells is very thick. Then the enzyme Rubisco, this one, is present only in bundle sheath cells. It is completely absent here. It's a very important point we have to remember. Then, in the mesophyll cells, there is another enzyme known as pepcase, phosphoenol pyruvate carboxylase. Phosphoenol pyruvate carboxylase. Simply we call it as pepcase. So pepcase enzyme is present in the mesophyll cells, but absent in the bundle sheath cells. So again I repeat, Rubisco is present in bundle sheath cells and pepcase is present in the mesophyll cells. So in the presence of pepcase enzyme, carbon dioxide fixation takes place or carboxylation takes place. This carbon dioxide combines with water, forms HCO3, and HCO3 combines with phosphoenol pyruvate in the presence of pepcase, and oxaloacetic acid is formed. Oxaloacetic acid is the first stable carbon compound formed in C4 pathway. So what is the first stable carbon compound? It is the C4 acid, oxaloacetic acid. This is the first step. And the enzyme used is pepcase. This oxaloacetic acid, synthesized here, is converted into another 4C acid known as malic acid and some other acids. This malic acid is transported from mesophyll cells into the bundle sheath cells through plasmodesmata. After entering the bundle sheath cells, the malic acid dissociates into carbon dioxide and pyruvic acid. Pyruvic acid is citric acid. Dissociation of carbon dioxide from malic acid is known as decarboxylation. D means removal. So in this way, carbon dioxide is released into the bundle sheath cells. So here carbon dioxide is coming in, it combines with phosphoenol pyruvate and it is transported in the form of malic acid into the bundle sheath cells. That means carbon dioxide is transported through these cells into the bundle sheath cells. Then the pyruvic acid goes back into the mesophyll cell, and from this pyruvic acid again PEP is synthesized. Phosphoenol pyruvate is synthesized by using some energy ATP. So in this way, ultimately PEP is resynthesized or regenerated. As RUBP is regenerated here, PEP is regenerated. So in this way, the cycle goes on. Then, what is the purpose of this cycle? It has great significance. The C4 pathway is costlier when compared with C3 pathway. More energy is spent in C4 pathway. Then why plants have adopted a costly pathway, that is C4 pathway, over C3 pathway. This pathway has many advantages. In the C4 pathway, photorespiration is completely prevented. I will come to the point later on. Photorespiration is one of the problem for C3 plants. In the case of C3 plants, photorespiration results in the loss of productivity. Nearly 1/4 of the productivity is lost in C3 plants because of photorespiration. But in C4 plants, photorespiration is completely prevented. So, the productivity is very high here when compared to C3 plants because of the absence of photorespiration. Spending of few more molecules of ATP is not a problem for these plants because the productivity rate is very high here. And these plants receive sunlight for longer duration because of their tropical nature. That's why the productivity is very high, and it doesn't matter if it loses some energy in the form of the cycle. Then, significance of C4 pathway over C3 pathway. I already have told these points. Carbon dioxide assimilation is greater in C4 plants than in C3 plants. So the carbon dioxide assimilation is more here, greater. Photooxidation is prevented. Here there is no photooxidation. Better adapted for environmental stress like heat and dry conditions. Utilize more ATP than C3 plants, but it is not a problem due to high productivity. Then, here the primary carbon dioxide acceptor is PEP, phosphoenol pyruvate, a 3C compound. But here it is the 5C compound RUBP. That is one main difference between these two. And that reaction occurs in the mesophyll cells. Then, now let us deal with photorespiration. I have told you this enzyme, Rubisco, ribulose, bisphosphate, carboxylase, and oxidase enzyme. C stands for carboxylation, O stands for oxidation. That means Rubisco is capable of participating in both carboxylation and also oxidation. It depends upon the availability of these two gases, of these two substances. If carbon dioxide occurs in high concentration, carboxylation takes place. If oxygen occurs in high quantities, oxidation takes place. So it depends upon the concentration of carbon dioxide and oxygen. It means Rubisco has reactive centers for both carbon dioxide and oxygen. In C3 plants, what happens is carbon dioxide and oxygen occur simultaneously, together. So what happens? Both carboxylation and oxidation will be occurring simultaneously in C3 plants. As a result, what happens? Instead of producing PGA, instead of producing 2 molecules of PGA, so that means, in other words, I will tell you, when Rubisco combines with carbon dioxide, what we got? We got 2 molecules of PGA. But when Rubisco combines with oxygen, what do we get? We get only 1 molecule of PGA and 1 molecule of phosphoglycerate. Phosphoglycerate. So instead of getting 2 PGA molecules, we get only 1 PGA molecule and 1 phosphoglycerate. This phosphoglycerate is a 2-carbon compound. That's why photorespiration is also known as a 2C cycle or 2C pathway. This 2-carbon phosphoglycerate is a wasteful product. It serves no purpose. In addition, it consumes ATP and it produces carbon dioxide, which is released out. So, photooxidation is a wasteful process where only one PGA molecule is produced and the other molecule produced is phosphoglycerate, which ultimately releases carbon dioxide by consuming ATP. So this is a wasteful process. But that wasteful process is inevitable here in C3 pathway. So the productivity is always low in C4 pathways, C3 pathway than in C4 pathway. Then let us see how this wasteful expenditure is prevented in C4 pathway. Here, carbon dioxide combines with PEP and oxaloacetic acid is formed. That oxaloacetic acid is brought into the bundle sheath cells. Then, I have told you the enzyme Rubisco is present in the bundle sheath cells and in the mesophyll cells peptidase is present. For photooxidation, this enzyme is required. Rubisco is required and that Rubisco is in these cells. Here what happens is when malic acid dissociates and carbon dioxide is released, it is released very close to this enzyme, Rubisco enzyme. As a result, Rubisco enzyme picks up carbon dioxide and there is no chance for photorespiration. In that way, photorespiration is prevented in C4 pathway. I repeat again. In C4 pathway, what happens? Rubisco enzyme is present in the bundle sheath cells and carbon dioxide is released very close to the enzyme. Because it is released very close to the enzyme, the Rubisco enzyme picks up carbon dioxide instead of oxygen. And in this way, photorespiration is totally prevented in C4 pathway. That's why the productivity is very high in this mechanism or C4 pathway or Hatch and Slack pathway. That is about Calvin cycle, Hatch and Slack pathway and photorespiration.